Let's see now some additional details about the adaptive immune system. And just to remind you, for the adaptive immune system, we have two types of cells. We have the B lymphocytes and the T lymphocytes, or the B cells and T cells. And we said the B cells are going to be involved in humoral immunity, whereas the T cells in cellular immunity. Both would need antigens, and therefore there is another group of cells for the adaptive immune system, that is the antigen presenting cells. We said those are the cells, they are going to hold the antigen, mainly for T cells to recognize it and decide if it's a self-antigen or non-self antigen. Those antigen presenting cells could be macrophages, dendritic cells, or even B cells, and those will show the antigen they need to show to the T cells on the MHC2 class. One thing about those lymphocytes we need to understand that they have kind of their own story, their own life. The B cells and the T cells, they have kind of different paths, although the basic concepts or the basic steps are the same. So for the lymphocytes, we said we have the B cells and the T cells. Both are produced, as we know, in the bone marrow. So production in both is bone marrow. That is where the mother cell produces them. But after production, they need to mature. What does mature mean? It means learn how to differentiate between self and non-self antigens. Because the whole adaptive immune system is about this, recognizing the self and non-self antigens. If it is a self antigen, they will leave the cell alone. If it's non-self, that means this cell need to be, taken down. For maturation for the B cells it happens in the same place, in the bone marrow. That is for the B cells. For the T cells, it happens in the thymus. So the T cells need to migrate to the thymus, and there they will start to learn how to recognize self versus non-self. Then the next step after maturation is activation. And here, for both of them, you can think of the lymph nodes as a site of activation. What do we mean by activation? Here, it means that they recognize a non-self antigen and activate or start adaptive immune response. Okay, so once they come in contact with a non-self antigen, that is the activation. And for those T cells and B cells, they might stay in lymphocytes like months or even maybe years before they get, activated. But they are there. And eventually, at one point, they will get activated. Okay, so let's try to understand now more details about the T cells. For the T cells, upon production or upon development, we have two types of T cells, either a CD4 or CD8. What are those? Those are receptors on the surface of the cell. This is a T cell that it might display CD8 or it might display CD4. Therefore, this will be CD4. This will be CD. As simple as we can explain it. So that is when they are born. They will go through maturation, and again in the thymus, they will remain as CD4 or CD8. But then, that is where they will develop. Upon activation, the CD4 will turn into T helper cell, whereas CD8 will turn into T cytotoxic cell. So the CD8 will turn into cytotoxic cell, the CD4 will turn into helpers. So that is one difference between the two. What does it mean, helper cell, T helper cell? It means it will help in activating or initiating adaptive immune response. From its name, it's helper. So we will see that it might help the CD8 and it will help the B cells sometimes. Okay, that's the T helper cell. The cytotoxic cell, from its name, it will kill the defected cells. What do we mean by defected? Maybe infected cells with viruses, maybe cancer cells. Any cells that the T cells or the adaptive immune response deemed to be inappropriate, deemed to be defected, deemed to be damaged, and we need to get rid of it because other problems might appear if it remains there, then the cytotoxic cell will go and kill it. So that is the difference in their activation and therefore in their function. But then there is a third difference. That is in terms the way they get activated, or like to be specific, if they can read MHC complexes. The T helper cell can read MHC2, whereas the cytotoxic cell, it can read MHC1. What does this mean? It means that the CD4, and we will see it, when it comes in contact with antigen presenting cell, it will bind to its MHC2. But the cytotoxic cell, or the CD8, when it comes in contact with antigen presenting cell, it will bind to the MHC1. Those cells, like if we go back here to activation, after activation, we need to add, they will get activated into T helper cell and here into cytotoxic cell plus memory cells. And the advantage of having those memory cells is in case we have second infection, those will get activated much faster. Okay, and we will talk about the second infection when we talk about the B cells. Okay, with that out of our way, let's go back to the mechanism of activating the T helper cells and the cytotoxic cell. And if you remember, we said T cells in general is cellular immunity. It means that it needs to come in contact with the antigen, and that antigen is shown on to it on the antigen presenting cell. So as we described earlier here, this is what's going to happen in the activating of both cells. So let's assume that we have a macrophage. This macrophage ate a cancer cell or a viral infected cell, developed antigens out of that cell, and now it needs to activate the helper T cell. We'll start with the helper T cell. So it's going to display on its MHC2. This is MHC2, a non-self antigen. And then it will secrete cytokines they are going to call a CD4 cell, a T CD4 cell. Where is this CD4 cell? It's here. Remember, we call it CD4 because it's not activated yet. This will get activated into T helper cell. So those cytokines are kind of a guide or telling the CD4 that there is a macrophage over here and please come and check it. So what's going to happen is this macrophage, again here, is still displaying the non-self antigen. So what's going to happen is we have the same macrophage here. The CD4 would come toward the macrophage. This is still CD4. And this CD4 is going to do two things. First one is going to recognize the non-self antigen. Okay, this is one here. Recognize non-self antigen. And then it has to recognize the MHC2. Recognize MHC. What is the point? The point is that this T helper cell, in order to get activated, it needs to double check on both. That the antigen is non-self antigen and the carrier of that antigen is MHC2. If either one failed, then we will not get activation. Think of it, activation is a very serious business. So we cannot have mistakes. And it is a serious business because if this get activated, if the CD4 get activated for the wrong reason, this might mean autoimmune disease for wrong reason. So both of them, both 1 and 2, need to be checked before the CD4 get activated. So once this is checked that the CD4 is recognizing that this is non-self antigen and the CD4 is recognizing that it is displayed on the correct holder or correct complex, then the CD4 will turn into T helper cell. What does the T helper cell do? It will start to secrete cytokines to alert other immune cells and kind of kick off the adaptive immune system. Okay, so this is the way the T helper cell get activated, by recognizing the non-self antigen on MHC2. Okay, so now we have the T helper cell. Now let's keep going and see how do we activate the CD8 cell, the cytotoxic cell. So we will use the same macrophage we have here. I'm just going to copy it and paste it. So now we have the CD4 cell. It got activated. So now it turned into a helper cell. So we said this T helper cell will start to secrete cytokines. We said to call other immune cells, right? Then what type of other immune cells? It's our CD8 cell. So what do those cytokines are saying to the CD8 cell? Think of it like, come to check this. So in this case, the CD8 cell would move toward the macrophage, and over there it will bind to its MHC1. So here we have MHC1 displaying the non-self antigen, and here now the the CD8 cell is going to recognize this MHC1. So it needs to recognize the non-self antigen, right? And it needs to recognize the MHC1. So what the T helper cell is telling the CD8 cell to come to check this and go to find it and find find it in the body. And that's why it has to see it on MHC1, because this MHC1, we said we will find it on all nucleated cells, right? So now the CD8 cell, instead of CD8, it will turn into cytotoxic cell. It will get off the macrophage, the cytotoxic cell now, and it has to find this non-self antigen on other cells. So let's assume it was cancer in the liver, for example. Those are the liver cells. All of them, they have MHC1 as nucleated cells. Some of them will display self-antigen, and some of them will display the non-self antigen. So this TC cell would come and check this non-self antigen, and of course it will check the MHC1. Is it self or non-self? In this case, it is a self, so this will pass. Here, it is a self, it will pass. Here, it's non-self. So the TC cell will come here and bind this cell and do exactly as we said earlier with the natural killer cell. So it will drill a hole in the plasma membrane, then secrete enzymes to destroy the cell. So eventually this cell will get eliminated. Why? Because it's displaying non-self antigen. Here it will pass because this is a self antigen. That's why we call them cytotoxic cells. So their activation is a very complex process. The T helper cells need to verify their activation, because again, if they got activated, they are going to kill other cells. And imagine again, as we said earlier, if they got activated for the wrong reason, that is an autoimmune disease, and that's a big problem. Okay, so that's for the activation of the T cells. I will stop here, and in the following video, we'll talk about the activation of B cells.